All right, guys. So last segment of 2.2, IQ 2.2 and podcast 2.2. I'm just on, still focusing on the central dogma, but just specifically now on translation. So as you guys have told me, part number 3, as you guys have told me that these are useful to you guys, what I wanted to mention before I dive into the content here is how you should be using these podcasts. I want to be very clear about something. My goal for using these for you guys was as a replacement for lecture. But also as a primer to get you started on to the main way that you should be consuming the content. So please don't rely just on the video podcasts as your only method of confronting and consuming the content of AP Bio. There's a lot of materials you guys know by now. And the class could be dangerously close to just me yapping at you the entire time, preaching at you about this is what AP Bio is. And I don't think that's the way that science is done. We need time to do science, which is doing activities in labs in class. So don't confuse just using these as the only thing that you should be doing. That would be just like listening to somebody giving you a lecture and then brushing it to the side and saying, okay, that's good. You also need to look at the book. You go through the IQ, go through the supplementary videos, and spend time with it on your own, okay? Read the text, take detailed notes with the IQ and the Prezi, watch those supplementary videos, ask questions when I ask if you guys have questions. Using this as your sole source just isn't going to work for the course. I want you to watch these first. I want to prime you up and get you thinking about some things and be able to explain some things to you. But doing these other things is absolutely necessary to your success. So I hope that you're doing them. And I just wanted to say that before we dive into the rest of the podcast here. Okay? So putting that aside, in this podcast, what are you guys going to be learning about? We're going to talk a little bit about Rnase versatility. So I asked you to look at the end of section 17.3 where it talks about alternative RNA splicing. And also I gave you guys something to look at in Bill Bryson's chapter 26, the Stuff of Life book. There's one specific section in there when I'm asking about the proteome where this is going to help you with that. So I'll explain that to start today. I'm also going to talk about ribosomes and what happens at the ribosome, how the genetic code is used to determine an amino acid sequence, which determines polypeptides, which then determines the three-dimensional shape of a protein. And then translation of mRNA into polypeptides. Final thing will be eukaryotic versus prokaryotic translation. All of this, again, covered in the book and covered in the IQ presentation and those supplementary videos. This is just your first walk through it. Okay, so let's go through it. We are at part 2 of protein synthesis. We talked about transcription. You guys asked some good questions today. Let's talk about the second part. So we are leaving the nucleus with our mRNA copy. After RNA polymerase has done its job, and we're going to try to turn that into, the eventual goal is that we're going to use that code and turn that into something tangible that we're going to call a polypeptide sequence, which is made out of amino acids. The first thing I wanted to discuss, though, is RNA, and how I would actually say that RNA is much more interesting than DNA. DNA just kind of sits there in the nucleus, especially in eukaryotic cells, and doesn't do much. I mean, it codes for everything in our bodies, but action-wise and moving around, it has to stay in the nucleus. RNA is actually much, much, much more versatile than DNA is. And it can fold into all sorts of shapes. So what this is showing here is like the beginning part of this central dogma idea where we have DNA, and that DNA is coding for a single-stranded RNA strand of one particular segment of DNA that we're interested in. But take a look at what happens here. Because of the single-stranded nature of RNA, it can actually fold up on itself and make some very specific shapes. And those things are called ribozymes. Now, these haven't been known about for a particularly long time. The person that did discover them, his name is Thomas Chick. He is a professor actually at the University of Colorado Boulder, and he still teaches a general chemistry course there. So possibly if you guys go to CU Boulder, you'll get taught your general chem classes by this guy, a Nobel Prize winner who discovered ribozymes. I put a video in the description, in the video category on the website about the discovery of ribozymes and how that was awarded the Nobel Prize. So take a look at that. But RNA, I would argue, is much more interesting than DNA because it can fold into these little shapes and it can act like a catalyst, which we usually call enzymes, which are proteins. Not always proteins, though. And this is a great connection to evolution when we talked about, well, RNA must have been the first information molecule. So RNA can hold information, but it also can fold into these shapes by hydrogen binding with itself. And we're going to look at one specific example here of how RNA will bind with itself. One great example is tRNA. So we have mRNA, which is our single-stranded copy used from the template of DNA. That's going to leave the nucleus and needs to be expressed into a polypeptide. Well, one very, very important other type of RNA is called tRNA. It stands for transfer RNA. This here is a 5 to 3' coded sequence of RNA, right? That just comes from DNA. But this tRNA is special. It will fold up on itself, which is represented by these lines here, these hydrogen bonds, where it's bonding with itself. And now look at the special shape it has. The special shape it has allows it to carry amino acids. It also allows it to have 3 exposed bases down here at the bottom in this loop called the anticodon. That's going to be really, really important later. So we've got these hydrogen bonds in this specific shape folding up. As simplicity's sake of a model, what you're going to see that as is it just looks like this. Let's use this as the model. We know that this is what it does. It's made out of code, and it folds up on itself and interacts with itself and uses hydrogen bonds to get its specific shape. But this is the model we're going to work with, where we've got this end attaching to a specific amino acid, and we've got this end, which is a specific anticodon. That's the symbol that's used in your book. Okay, so that's one great example of RNA being much more versatile than DNA. Okay, here's your whole picture. We've talked about transcription, and we've talked about RNA processing, so post-transcriptional. So adding a 5' cap and a poly A tail, and cutting out introns, and putting in exons, and splicing them together with the spliceosome. One extra detail when I asked you guys to read that section of the book that RNA does that's really interesting is called alternative splicing. Take a look at that and then ask me questions, please, about it tomorrow if you have some. It's a really, really interesting part of what RNA does. So here you go. You got RNA polymerase making its copy. We've got our mRNA strand. That now needs to leave the nucleus. Notice we've got the poly A tail and the 5' cap labeled here. mRNA is leaving, and it is going to try to find this guy. Now, this is not to scale. Ribosomes are super, super tiny. They're around the endoplasmic reticulum out into the cytoplasm. We've got this protection here as all sorts of chemistry is happening out there in the cytoplasm. Protected, ready to go. Now we just need to find the ribosome, right? The first thing that needs to happen is something called amino acid activation. That is, you can see all these little purple shapes in this image here. A bunch of amino acids floating around. They need to bind to the correct transfer RNA. So there was that spot on transfer RNA where amino acids can bind. That we call that just charging up or being activated. That tRNA is going to grab the correct amino acid. And then that tRNA needs to make its way down here to the ribosome, which is what we can see right here. That process is called translation, right? So here's the charging up, and that is using a specific enzyme called amino acyl tRNA synthetase. Do you need to memorize that? No. But this is the enzyme that does that. We'll get much, much more in enzymes in biochemistry, which are these amazing little chemists that do one job really, really well, which means they have to have one shape in order to do their job, their specific chemistry that they're doing. Notice the specificity of this shape. Where this amino acid might be shaped this way, there might be other amino acids that are shaped completely different. There are 20 amino acids, all shaped different. This enzyme will only bind that amino acid, and it will only bind this particular type of tRNA because of a little code down here it has on the bottom that will charge it up. And now this job, the amino acyl tRNA synthetase, all it does is it takes the correct amino acid and it joins it to the correct tRNA based on the anticodon it has down at the bottom of that tRNA. It is now charged up, which just means when we say charged up, that the tRNA now has the correct amino acid with it. Now that's super important as we go on to the process of translation where we try to use mRNA and tRNA together in order to synthesize and put together these amino acids that are sitting there on top. Okay? So there's amino acid activation. We've got the correct tRNA matched up with the right amino acid. Now we need to go down here to the ribosome. Here is a picture of what's going on in the ribosome. Again, you've got all your amino acids. They're getting charged up here, and now they're going to make their way down. You've got your mRNA strand here. Remember what's on the mRNA strand is you've got this 5' cap. You've got these regions that code for nothing. What we want to get to is the exons. In order to know where to begin, the mRNA strand needs to have some sort of start signal. So it has a start codon, which is actually going to code for the amino acid methionine. Once methionine is reached, the start codon is there. It says, okay, let's begin transcribing, in which it will go through this particular process here. We've got 3 different sites in the ribosome that we designate the E, the P, and the A site. What happens in the A site is you've got the charged tRNA coming in, And notice it's being fed through the ribosome in this direction. So we're going 5 to 3 in this direction. And into the A site, this anticodon is going to bind onto the correct codon, the 3-letter sequence that's actually on the mRNA strand, and it's going to bind to it, and it brings its amino acid with it. Then in the P site, what happens is you've got a transferring over of the amino acid to this growing polypeptide chain. Polypeptide chain is just of a bunch of amino acids linked together. That's all that's happening here. So in the P site, we've got this amino acid binding now to this previous chain that we have of this polypeptide. That requires a little bit of energy, which is just showing here with GTP. Peptide formation is now being formed. Now what we've got is a shuffling over of the tRNA. So the next tRNA needs to come in and have space in this A spot. So everything needs to shift over. That's what's being shown here. That we've got now this old tRNA, which was this one eventually coming in, is now shifted over to the P site. It holds on to the growing polypeptide chain. The old tRNA now needs to be back out into the cytoplasm to be charged back up by its enzyme and grab this amino acid, which is now empty, so it gets ejected out of the E site there. And this process goes over and over and over again until you reach one of 3 stop codons. It says, okay, stop the process of translation. We have our finished polypeptide. When that polypeptide is complete, it will fold up into a very, very specific shape, which I'll show you here in a second, one really good example. This is something I briefly wanted to mention also. That's the redundancy in the genetic code, basically meaning that the genetic code is interpreted as a series of codons, 3 letters at a time, which are going to make and code for a specific amino acid. There's 64 possible codons, which code for all 20 amino acids, along with 1 start codon and 3 stop codons. So that means that if I wanted to code for something like, let's look at arginine down here, I could have CGU, which means arginine, but let's say that I have a single nucleotide polymorphism, an SNP, a SNP, that comes along and it changes this U randomly. We have a mistake, maybe due because of during DNA replication, something got screwed up, and we have a C there instead. Well, the redundancy of the genetic code says that's not actually that bad of a situation, because what we actually have is it's still coding for arginine. And when you get down to the end of it, the end of translation, all that really matters is, do we have the correct amino acid sequence? If we have the correct amino acid sequence, we're going to get the correct shape of the protein, which means we're going to have the correct job happening. So one thing that you should be able to do is read this wheel here and determine, well, what amino acids are going to be made using this particular sequence. Here's one example. Please stop and pause the video and see if you can't do this on your own. I'm going to go through it quickly. If I have a copy of DNA with these letters here, one thing I should be able to do is take a look at, well, where's my template strand and where's my non-template strand? What am I trying to make a copy of? This is what I'm going to try to make a copy of, TAC. So my mRNA strand, this is transcription that I'm trying to do. I'm going to make a copy, and I would start with AUG, the complements, GCC, AUA, UUU, GAC, and UAA. That's my 5' to 3' copy, in which it's going to enter the ribosome and start being read here. So the amino acids that would get made, here's AUG. If I'm going to use this, there's A for the first part, the 5' end. There's U and there's G. So we would be using methionine. That would be our start codon, methionine. I'm just going to use the symbol M to start it. We would then go on, pause the video real quick here and see if you can get the amino acid sequence. Okay, welcome back. Hope you got the amino acid sequence written down. The sequence should go MA, I, F, D, and then you finally have the stop codon that says, okay, let's disassemble the ribosome and let's fall off, because now we've got our polypeptide chain here, right? When you've got your polypeptide chains, what's going to happen is they're going to fold on themselves. We go into this in much more detail in biochemistry into why they fold into 3-dimensional shapes. Basically, it's interactions of their own chemistry, sort of like how RNA interacts with itself with its own chemistry. So do these polypeptide chains, and they'll fold up and they'll make all sorts of these crazy folds. This is an example of a polypeptide, 4 polypeptides interacting that make up the protein we call hemoglobin. Hemoglobin, this is inside of a red blood cell. This is a artist rendering of what hemoglobin looks like. And then this little orange bit is molecular oxygen binding to the specific shape here of hemoglobin. Right, so oxygen is being carried around if hemoglobin is the correct shape. So here are the red blood cells, and those are normally shaped good red blood cells. But what would happen if we had a single nucleotide polymorphism that happened somewhere in the process of transcription and translation, and we had the wrong amino acid sequence put together, which means we got the wrong folding of the protein, and it turned into a sickle shape? And then would anything that we've discussed in evolution being able to enact upon that? Possibly. So sickle cell anemia never goes away. That is a process called mutation, which gives you variation.